So let's talk about, we're going to continue on in the urinary tract, or the urinary system, and since we're discussing about what occurs in the nephron system, and so we're talking about resorption. What materials are getting resorbed? Remember, there's three things that occur along our tubular system. Okay, you have filtration. All right, that's when everything gets knocked out of the glomerulus, all right, and into the tubular system. All right, then you get reabsorption, all right, when we reabsorb all the nutrients or the things that we want to reabsorb, primarily in the proximal convoluted tubule, okay, but you'll get resorption in the loop of nephron. We'll talk more about that today. And even in the distal convoluted tubule and the collecting tubule near the end of our nephron system. And then the last one is secretion, all right, because sometimes we'll resorb something, all right, but then we're like, we don't want it. We're going to secrete it back into the tubular system and put it back into the urine because, say, your blood pressure has risen, your blood volume is quite high, we need to get rid of some water, let's get rid of the water to decrease the blood volume. All right, so we'll secrete the water into the tubular system and into their urine, okay? So when we talk about those systems, we're just going to be, or those three processes, we're focusing right now on nutrient resorption, okay? So this is very important. When we talk about a specific nutrient, each nutrient has its own specific transport protein. All right, this brings us back to the wonderful days of chapter 4 when we learned that there was different types of transport proteins. All right, you had your channels, all right, that's a type of transport protein, all right. You also had your pumps, that's another type of transport protein, and then you had your transporters, all right, and we didn't really talk too much about the transporters, but we're going to talk about them today, all right. In fact, when we talk about glucose transport, we're going to talk about a structure called a symporter, okay. Sym is two, you're going to be transporting two things, all right, in the same direction. S is for symport, S for same, okay? So keep in mind, when we're talking about transport protein, remember, proteins have tons of different functions, all right? So these are just a couple. I hope you can read my writing there. So let's talk about glucose transport. It's a crucial macromolecule, okay? It's a carbohydrate, all right? Your body loves it. The brain craves it. It's its primary source for energy. Okay, so I'm going to come back to this slide. I love pictures, especially this chapter. We're going to be needing to see pictures quite a bit. All right, so I'll tell you the story, and then I'll bring you back to that slide. All right, so here we're looking, all right, into our tubular system here. This is where we're at. We're in the proximal convoluted tubule here. All right, so basically, all right, I'll tell you the outcome of the story. All right, the hero wins at the end. 100% of the glucose is resorbed back into the blood. Okay, so glucose is a fairly small molecule. It'll move through the filtering system or filtration membrane in the glomerulus there. Not to worry, in a normal, healthy individual, we're going to resorb 100% of that glucose, which is good. We want that, okay? That's why you shouldn't find glucose in your urine in a healthy individual, all right? So here's what happens. In the tubular fluid, all right, outside the cell, remember this from chapter 12 and 10 when we talked about the ion concentration inside of cells and outside of cells. Sodium is always high outside the cell, okay? So what will happen is we have this wonderful symporter, all right, that is going to allow sodium to move down its concentration gradient. When we're dealing with concentration gradients and things that move passively down a concentration gradient, we're dealing with passive transport. Doesn't require any energy, okay? So as sodium moves down its concentration gradient, all right, it's going to generate energy, kinetic energy specifically. Well, that's enough energy now that our glucose molecule can piggyback on top of that. And it's going to go through the same transport membrane, same transport protein, excuse me, okay? And that is what we call a symporter. So we have two different substances going in the same direction, all right? One is going to go down its concentration gradient. That's going to be sodium. Glucose is going to move up its concentration gradient. So now you've got sodium here in the cell. We're going to leave that alone. We're done talking about that for now, okay? So our glucose now is in a higher concentration here inside the cell compared to the interstitial fluid on the other side of that cell membrane. All right, this is the luminal surface here. All right, that's going to be in the proximal convoluted tubule. And then you've got your what we call the basolateral membrane surface here. That's the bottom of the cell. All right, on the other side of that is our blood vessel. Okay, so the peritubular capillaries will be sitting over here. All right, so we have high concentration of glucose here, low concentration of glucose here in the interstitial fluid. Well, it's easy. All right, now glucose is going to move through with this, it's called the carrier protein. All right, it's going to move through this protein, this glucose uniporter. Uni meaning one. One thing is going to be moved through, and glucose goes from an area of high concentration to low concentration. It moves right across and goes right back into the blood. Okay? As long as there's not too much glucose, you're a relatively healthy individual, all that glucose is going to go back into the blood. Listen, you see this term here, diffusion. All right, diffusion is a passive process. There's no energy required for this process there. All right, so basically, in words, that's that picture that I just showed you. This is everything here in words for that. Okay, so sodium, as it's moving down its concentration gradient, generates energy. Well, that's perfect because glucose now can move up its concentration gradient into that tubule cell. Okay, from the energy that the sodium generated. All right, and then it moves into the cell, and on the other side of the cell there, the basolateral membrane surface, all right, it's moved out of the cell through the uniporter, okay? It's going down its concentration gradient, and it's going to wind back up into the peritubular capillaries. Do y'all feel pretty comfortable with that process? I know this is kind of bringing back some concepts that you might not have used in a while. Concentration gradients, all right, diffusion. All right, there's two passive processes, okay, for transport. You've got diffusion, all right, you have simple diffusion, and you have facilitated diffusion. Simple is just essentially, all right, stuff moving across the plasma membrane on its own. Doesn't get any help. If it's facilitated diffusion, it's getting some help. Well, what helps it? A channel, all right? A leak channel, for example, that we saw in the neurons, all right? Or this channel here that we just saw, the symporter, all right? That's facilitated diffusion. Right? Still, it does not require energy. Right? We don't need ATP. It does generate energy, and that's good because as that sodium is moving down its concentration gradient, it generates energy for glucose now to use because it's got to move up its concentration gradient. Think of it like rolling a boulder up a hill. You've got to use energy for that process there. Okay? And then eventually now glucose will move into the blood because it's going to move back down its concentration gradient through the uniporter there on the basolateral surface of that cell. All right, so now that's how we resorb 100% of all that glucose that got filtered out, all right, into the filtrate and then moved into the tubular fluid here, the tubular system. It moves right back in. So that should be 100% that gets resorbed there. All right. So since we're on the subject of substances that are completely resorbed, let's talk about protein. All right, remember, when we talked about protein before in the filtrate, we said large protein molecules won't cross over, all right, or very rarely, or if there's some sort of pathology going on. It's going to be the small protein and the intermediate size ones that will cross over, all right? So we should not really see large protein molecules there. All right, but what will happen is, same scenario, all right? We're going to deal with the proximal convoluted tubule here. So all those proteins, okay, are going to be in the tubular fluid, all right? You have those tubular cells that are going to take that protein into the cell through two processes, pinocytosis and receptor-mediated endocytosis. Okay, those are two processes in which you have something that's outside the cell, and the cell essentially gobbles up whatever's outside and pulls it inside. That's what we're going to see here. So the protein is going to move across that luminal surface there. All right, it's going to be taken to the inside of that tubular cell. Then that tubular cell is going to use lysosomes, which have digestive enzymes inside. Okay, they're a membrane-bound organelle. Or peptidases. Well, we remember peptidases from when we were doing chapter 26 and talking about enzymes that break down, all right, peptide bonds. That's what we're going to see here. These peptidases and these lysosomes will basically digest these proteins and break them down into those amino acids. And how nice. All right, now we have something really small, and we can move those amino acids back into the blood through facilitated diffusion. Well, what's facilitated diffusion? It's a passive process. All right, that means we have some sort of channel there, all right, or gate that's going to let that substance move across. But again, it's passive. So that means it involves, all right, a concentration gradient. So that means that we have to have a lot more amino acids inside the cell than in the blood. And that's no problem, because we got that. All right? So this is another example of 100% complete resorption. And that's going to be our proteins. Again, you should not see proteins in your urinalysis. You go and pee in a cup, and they check, all right, your urine for sugar or blood or white blood cells. Proteins should not be in there. Now, there are some situations where you might have some trace amounts, okay? But normally, you should not see proteins in your urine there, okay? So now we've talked about substances that are going to be completely resorbed. Now we're going to talk about substances that are going to be regulated in regards to resorption. What does that mean? That basically means if you have a lot of something, okay, you're going to get rid of it. Certain situations are going to alter the amount of whatever you're resorbing. It's either going to increase the resorption or it's going to decrease the resorption and thereby increase its secretion, all right, and put it into the urine. All right, so let's talk about sodium. Very important ion. Okay, we know the importance of sodium, all right, when we talked about certain physiological processes in our body. All right, so pretty much the majority, if not all of sodium, is going to be resorbed from the tubular fluid. And you kind of saw that a little bit when we were talking about glucose here. Okay, so we're going to see resorption occurring, all right, across the nephron, and then again, primarily in the proximal convoluted tubule. That's where everything goes on. Proximal convoluted tubule does a lot. All right? So we already remember, if you had me in 210, I had a saying, banana floating in the ocean. All right? Think of a banana floating in the salt water. Banana's high in potassium, so that's the inside of the cell. High in potassium, outside the cell, high in sodium and chloride. All right? So therefore, sodium has a low concentration inside the tubule cell. So that means we have a concentration gradient set up. It's high outside the cell, low inside the cell. So where does it want to go? It wants to go inside the cell. We just need to give it a means to get it in there. All right? We have to somehow get it across. Okay? So we'll see, all right, the difference here, all right, with the tubule lumen here, sodium is going to move across, all right, the tubule membrane there, the luminal membrane, all right, and it's going to enter into the tubular cell, moving down its concentration gradient, okay? Now, once it's inside, it's where it wants to be because it has a low concentration gradient, but outside the cell has a higher concentration of sodium. So what we have to do to get it out of the cell is we got to force it against its concentration gradient. Well, we got just the tool for that job, pumps. Pumps, they're active transport processes, and usually active transport mechanisms require energy, ATP. Sodium potassium ion pump in the neuron. All right, those are always pumping away, trying to establish a concentration gradient in the neuron. Right? They actually utilize 66% or 2/3 of the total energy expenditure of a neuron is just spent operating this pump. Well, guess what? These sodium potassium pumps, which are found in the basolateral membrane there, that's the membrane that's closest to the bloodstream, all right, is going to do the same thing. It needs a lot of energy, all right? But it's because of this pump, all right, it pumps sodium out of the cell, potassium into the cell. It keeps sodium levels low inside the cell on purpose. Okay, so it establishes that concentration gradient. So we can get sodium to flow from the tubular fluid into the tubular cell. And then we'll pump that sodium out of the tubule cell into the interstitial tissue space or fluid space between the tubule cell and the blood vessel. And then it'll move across into the blood vessel. So there's a couple substances that are going to play a role, all right, in regards to the reabsorption process here, all right, and those are going to be hormones, all right, aldosterone and ANP, all right, that one, remember that hormone, the atrial natriuretic peptide that is produced when we see an increase in stretch on the atrial walls in the heart because the blood pressure is increased. Right, and then we're going to make some changes in the kidneys, how much urine production we're going to cause. All right, we're going to talk about that now, okay? All right, so here's the thing. I've got a couple slides with pictures here, so you're going to see me go back and forth, all right? First of all, here's pretty much what we're looking at here for sodium resorption. Most of it is going to get resorbed here in the proximal convoluted tubule, all right? A significant amount will be down here in the nephron loop, so that's 90% there. All right, the remaining amount, all right, in the proximal the distal convoluted tubule, the collecting tubule, which is over here, and our collecting ducts, all right, that is going to be regulated by hormones and body needs. Okay, so that's where the regulation is going to occur. We'll discuss that here in a moment. All right. So just a quick review as to what aldosterone does. We know where it's produced, all right, in the adrenal cortex. All right, but aldosterone is actually going to cause protein synthesis, which means we're going to make some proteins. All right, well, what proteins? We're going to make sodium channels and sodium pumps, or excuse me, sodium potassium pumps. All right, so this hormone is going to upregulate protein production for these pumps, which will help to facilitate the movement of sodium in these cells here. Okay, we're going to stick them in the plasma membranes. You need to know these cells, the principal cells. All right, we talked about the principal cells briefly in chapter 24 of the lab portion of this class. They're located in the collecting duct. Okay, that's the last part of this whole system, the collecting duct. Okay, and their job is to increase sodium resorption, which means you'll have less sodium in the urine. Now, guess what? Here comes the follower. Remember, what does water do? It's the follower. All right, it's easily manipulated. You just have to entice it with stuff. You entice it with some sugar, like a little bit of glucose. It follows the glucose. Sodium, same thing. All right, water will follow sodium. Okay, so that means if I increase the secretion or excretion of sodium into my urine, water will follow it, and it will increase water in my urine, and vice versa. If I increase the resorption of sodium into my blood, well, then the water is going to follow that, and it's going to go into the blood, which means less water enters into your urine. Okay? So aldosterone is if it's going to increase sodium resorption, that means it's going to decrease your urine output. You'll have less sodium in your urine, therefore you'll have less water in your urine. You guys see that? Kind of following me? A little review from chapter 17, we were talking about aldosterone. All right? Atrial natriuretic peptide, we talked about this at the beginning of this chapter. All right, we saw what it does to the glomerular filtration rate. It increases the glomerular filtration rate. Well, how does it do that? Remember, it vasodilates the afferent arteriole. That's the artery that leads into the glomerulus. So that allows more blood flow, which means more filtrate. It also, all right, decreases the production of renin. And renin, when it gets converted to angiotensin II, will stimulate the mesangial cells to contract and squeeze those filtration slits closed. Well, guess what? We've inhibited that. That doesn't work anymore. So now the mesangial cells are nice and relaxed. Those filtration slits are wide open, so we can filter more stuff out through the glomerulus. All right? So what we see here with the atrial natriuretic peptide here, it's going to inhibit the resorption of sodium. Okay, so you've got aldosterone, which increases the resorption of sodium. Well, guess what? Atrial natriuretic peptide is going to decrease it. Because why? If AMP is getting produced, that means your blood pressure is too high. The stretch of your atria is too much. Your blood volume is too much. We've got to decrease the blood volume. How do we do that? Let's get rid of water. All right? How do we do that? We inhibit the resorption of sodium here in the proximal convoluted tubule and in the collecting tubules. All right, which means we're going to excrete more sodium in our urine. Well, guess what? Water's going to follow it. So now we've increased more sodium in the urine and more water. All right, and in addition to all that, AMP also inhibits the release of aldosterone. So one regulates the other. That's nice. That's good. All right, so let's talk about some of this stuff. All right, I already showed you that. Okay, zoom in. Now I'm going to show you in pictures what I just told you in words. All right, so here we go. We're in the proximal convoluted tubule. We have a lot of sodium here, okay? So we have our sodium channel here, no problem. It's facilitated diffusion, which means the sodium is going to diffuse down its concentration gradient, area of high concentration to low concentration, passive process, cool, no problems. Goes into the cell. All right, now, all right, we're in the cell, okay, and we have a low sodium concentration in the cell, and we want to move it out, but we have a high concentration of sodium outside the cell. So we need to move it against its concentration gradient. No problem. Got just the tool for that job. Sodium potassium ion pump. It's an active process. It's going to take some energy, but then again, all right, I wouldn't expect anything different. So we're going to move that sodium out of the cell against its concentration gradient, but at the same time, we're going to pull potassium back into the cell. Yeah, that's all we're doing. All right? We're just moving it across, balancing it out. All right? And then that sodium then enters back into the blood, all right? And then, of course, keep in mind, water follows it. Okay, so both sodium and water will go back into the bloodstream. And so pretty much not all the sodium, but about 65% of it is resorbed in the proximal convoluted tubule back into the blood. So we still have another 35% to deal with. All right? Okay. So now we're going to not fast forward, we're going to sprint to the end of our tubule system where the distal convoluted tubule, the collecting duct, and the collecting tubule is located. That means this is where we're at now. We're over here. Okay, we started here, that last slide. All right, now we're going to the end. Okay, so here, now we're going to be dealing with what we call principal cells. Okay, so these principal cells now, from aldosterone, okay, because let's say now, all right, you're dehydrated, okay? So you're dehydrated, your adrenal cortex is starting to secrete aldosterone because it wants to conserve water, all right, and it wants to decrease the amount of sodium that it's getting rid of, all right? The aldosterone here, you can see, here's our aldosterone, all right, it comes in and it starts to put into the plasma membrane more sodium potassium ion pumps and more sodium channels, okay? The sodium channels go on the luminal surface. This is the tube right here with all our fluids in it, all right? And then the sodium potassium ion pumps go on the basolateral side, okay? So here we're going to see, all right, because we have the sodium potassium ion pumps and this area here in the sodium channels, we're going to see, all right, sodium in its high concentration outside the cell moves across to into the intracellular space. The pump will move that sodium out. All right, and as the sodium moves across and back into the blood, here's water. Water follows it. All right, we'll talk about these structures here, aquaporins. They're just water channels. That's all it is. Okay, we'll talk about what happens, how we put more water channels in here in a second. All right, so notice, keep in mind, as sodium is going to be moving into the cell and out of the cell through the basolateral membrane here, potassium is going to be doing the same thing, but in the opposite direction. It's going from the basolateral surface into the cell and then back out of the cell. Okay? So aldosterone is going to help facilitate that. Here's one, and we'll talk about this here in a few moments. All right? Whereas aldosterone is going to increase the resorption, all right, of sodium and water. All right? It's also going to increase, all right, the secretion of potassium. You need to know that, because potassium levels play a high role, not a high role, that's a horrible word, a much more predominant role in determining the amount of aldosterone being released. In fact, high potassium levels, all right, will trigger, all right, the production of aldosterone much more so than your hydration levels. Potassium, yes, yes. We'll talk about that in a second here. Okay. So here you can see, we talked about sodium already. Well, water is going to be very similar, all right? So we call it obligatory water resorption because it's following the sodium here. So that water follows the sodium. We have 65% resorption of sodium in the proximal convoluted tubule. So is water because it follows the sodium here. We get a smaller amount here in the nephron loop. Again, we'll talk about that in just a little bit. All right. Instead of having 25% like sodium has in the nephron loop, we're only seeing about 10% of water resorption in the nephron loop. Part of that is certain parts of this loop are impermeable to water. Okay, so it doesn't have as much ease in passing through. Okay, but then again, when we get to the distal portion of our nephron, all right, structure, we'll start to regulate water resorption depending on some of the circumstances of what hormones are being produced, what are the body's needs, and whatnot. All right, so here now in the collecting duct and the collecting tubules here we're going to see how water, oops, where are we? Oh, that's too big. All right, same cell, principal cell, all right. Water can move through these aquaporins, all right, but also water can move right in between the cells. All right, we call that paracellular transport. Okay, it doesn't have to go through the cell, it goes in between. So water will move through the cells, all right, or excuse me, in between the cells. It'll move through the cells when we have these aquaporins in place. So ADH, that antidiuretic hormone, all right, that's produced in the hypothalamus, released by the posterior pituitary gland, all right, this hormone is going to increase these aquaporins, and the more aquaporins we have, all right, the easier it is for water to move across, all right, the cells that line the collecting tubules and the collecting ducts. So what happens? ADH increases or upregulates the synthesis of these aquaporins, and the cell will just insert them into the plasma membrane, and then water can freely move across. All right, now again, water is still obligated to move via osmosis. Remember that one, right? That's another passive process. And osmosis is governed by water moving from an area of high water concentration to low concentration through a semi-permeable membrane. Well, here's the semi-permeable membrane, plasma membrane. Okay? So think of it also like this. Water is going to go from an area of low solute concentration to an area of high solute concentration. So that's why when you're getting rid of sodium and resorbing it back into the bloodstream, water will follow it. Okay, it's going to go to that area that has the higher solute concentration. Conversely, all right, if you have a higher solute concentration, you have to have a lower water concentration. So it's going to flow down its concentration gradient. But that's just a review from last semester. All right, questions so far? All right, so we talked about sodium resorption. Okay, now we're going, and water. Now we're going to talk about potassium. This is another process, though, that is regulated resorption. Okay? So we're going to see, all right, how potassium is resorbed and how it's secreted. Okay? Because it undergoes both of these processes. Okay? It's a wide range, but 60 to 80%, all right, is resorbed in the tubular fluid when we're talking about potassium. And a big player in this, and what determines how potassium, all right, is going to be resorbed or secreted is sodium. Okay? Sodium. Same type of scenario here. All right? So we already know all this. Okay? Sodium is resorbed across the luminal membrane. Water is going to follow it. Now, what happens is when water leaves the tubular fluid, that tubular fluid increases its solute concentration. All right? Thereby, we have just created a concentration gradient differential between the tubular fluid and the interstitial fluid. All right? So what we're going to see here now is potassium is, of course, going to move down its concentration gradient. All right? From the tubular fluid, all right? And it's going to then move, all right, into the interstitial fluid. But guess what? It does it paracellularly. It goes in between the cells. It's like the heck with going through the cell. I'm going to go between. Why not? Okay? So we'll see it move. Hold on, I got a picture of it. No, I don't have a picture of it. That's later on. All right. We'll also see it get resorbed in the thick segment of the nephron. Okay. But here's the important one, this last portion right here. This is the important part, because that's usually where we're going to see the regulation occur. Okay. So we're going to go all the way to the end part where the collecting tubule is. Okay. And we're going to deal with these two cells, intercalated cells and principal cells. Well, I've just been talking about the principal cells. Those are the cells that were influenced by, all right, antidiuretic hormone and the aldosterone. Okay. Those hormones there, all right, we started putting more transport proteins into the plasma membrane. All right, well, easy to remember. Intercalated cells are only going to resorb potassium. That's all it does. All the time. All the time. Just how sodium-potassium ion pumps in a neuron are always pumping sodium and potassium across the plasma membrane, these cells are always going to be resorbing potassium. Okay, so while all this other stuff that I've been talking about is going on, and it varies depending on what hormones are present and what's going on, all right, with your hydration levels and other factors, all right, this goes on. Like the sun will rise and it will set, this happens. Okay, now what will change is what the principal cells do. Okay, they're going to secrete potassium, but it's based on, all right, how much aldosterone is present, because the aldosterone is going to stimulate the principal cells to secrete potassium. I'm going to come right back to this slide. Let's go back in time. Here you go. This is what I was talking about before. All right, don't pay attention to the sodium. Okay, you can see as we move, if we put more of these sodium-potassium ion pumps in the basal lateral membrane here, all right, if we start off with 1 and then we got up to 10, let's say, well, that means we're pumping out 10 more sodium compared to 1 sodium, because we have 10 pumps, but that also means we're going to pump into the cell 10 more potassium. And that potassium is going to cross over and get dumped into the tubular fluid. All right, and aldosterone does that. Okay, so aldosterone is going to, so the more aldosterone is present, the more potassium will be secreted. Like I was saying before, all right, the most powerful stimulant for aldosterone production is potassium levels. A lot of bad things can happen if you have elevated potassium levels. Okay, you can have some bad problems with your heart. Okay, you start to get your potassium levels askew, all right, you can have arrhythmias, all right? When you start to have arrhythmias, then that's a scary situation because you can have some other pathologies that go along with that. So that's why it's really important that we regulate our potassium levels, right? And aldosterone will do that. So elevated potassium levels, more aldosterone gets produced. If more aldosterone is getting produced, that means the principal cells are going to secrete more potassium. All right, how do they do it? They just put more potassium and sodium ion pumps into the principal cell's membrane, and we can move more potassium into the principal cell and then into our tubular fluid. And we dump it into the urine. There are certain diuretic drugs. Spironolactone is one of those. It's a steroid-based drug. It's what we also call a potassium-sparing diuretic. And so it prevents, because aldosterone is a steroid, all right? And so the spironolactone prevents potassium from being secreted, all right, when you are trying to decrease blood pressure. You give someone a diuretic, what are you doing? You're making them go pee-pee more. And if they're producing more urine, that means we're pulling more water out of their blood, which means we're decreasing the blood volume, which means we're decreasing the blood pressure. Okay? So here we're going to see it's a pretty wide range here. 60 to 80% of the potassium will get resorbed here. All right, in the proximal convoluted tubule, we'll get some resorption of potassium here in the nephron loop. But what we'll see, all right, we'll see quite a bit of potassium being either resorbed or secreted here, all right, in the collecting tubules and the collecting ducts, depending on what is going on with, all right, aldosterone levels. That's going to upregulate or increase the potassium secretion there. All right. Let's talk about a couple other substances, calcium and phosphate. All right, we've talked about this a little bit back and forth in other chapters. All right, chapter 7, we were talking about bones, and we talked about the presence of PTH, parathyroid hormone. We also talked about that in chapter 17 at the beginning of this semester. All right, we'll see, all right, how certain hormones are going to affect the secretion, the secretion excretion of calcium and phosphate. First, all right, when we talk about what's going on with calcium and phosphate, 60% of what's in your blood is going to get pushed into the filtrate. All right, the remaining amount won't because it's bound to calcium-carrying proteins like calmodulin and calsequestrin. It's a big molecule, so that won't get filtered out. It's got calcium attached. When you go get your blood work done, and I know if I said this before, but I'll say it again. When you go to the doctor to get your blood taken, all right, and they take the blood and you look to see how much calcium you have or sodium and all that, all right, that's the free floating calcium. That's just what's floating out there. Well, guess what? All right, 60% of that is going to get, that will go and get filtered out. Okay. 90 to 95% of that is going to pass right through the glomerular capillaries. So most of that calcium is going to get pushed out and get filtered. Okay? Now, what the nephron does with that depends on what your body's needs are going to be. So, for example, if your blood calcium levels are low, that means we want to save calcium. We don't want to get peed out. So parathyroid hormone gets secreted, okay? And so we need to increase our blood calcium levels. So what we'll see is we're going to stimulate and increase the amount of calcium resorption. In this case, that's going to occur in the distal convoluted tubule, okay? So we're going to really work hard to soak up and reabsorb as much calcium as we can, because our blood calcium levels are low and we need to increase them. Okay? What we'll see here also in the nephron is that we're going to inhibit, that's phosphate, we're going to inhibit our phosphate resorption. Okay? We want to get rid of phosphate. Okay? Because that means if we don't have phosphate floating around, it's that much more difficult for calcium to hook up and make hydroxyapatite, bone salts. Okay, because we're missing an ingredient for our bone salts. We don't have our phosphate molecule for that. So we're going to get rid of the phosphate in the urine. Okay, and therefore that means that calcium doesn't really have a home. That's okay because we don't want it to go home. All right, one of its homes is bone. All right, we want it floating around in the blood because our blood calcium levels are low. Okay, so we'll decrease the amount of calcium that's going to be put back into the bone matrix there because we don't have phosphate. All right, so therefore it'll stay in our circulation and that will increase the calcium levels. Simply, all right, by increasing the amount that we resorb in the distal convoluted tubule and by getting rid of phosphate, dumping that into the urine. All right, so here you can see, all right, parathyroid hormone, all right, is going to inhibit phosphate from being resorbed here. So it's going to work its way through the nephron system and it's going to get dumped into your urine. All right, well, as that tubular fluid moves through, we're going to resorb as much calcium here in the distal convoluted tubule to make sure that the amount of calcium in your urine is really low, and we start to work to increase the calcium levels in the blood. Okay, still staying on. I know I keep hitting you with this stuff, but now we're going to talk about, oh yes, I get to write out, well, I'll try to write it out. No, I won't. All right. Let's talk about acids and bases. That's everybody's favorite subject, right? Does anyone remember what the normal pH value of your blood should be? The range.
Between 7.35 and 7.45.
Bingo. Yep, that's the range. So normally you're looking at about 7.4, all right, is what you want the pH value to be. Now remember, pH is a scale from 0 to 14, and the lower it is, the more acidic it is. The higher, all right, the value of something is, the more basic or alkaline it is. 7 is neutral. It falls right in the middle. All right, hydrochloric acid, the acid that gets produced in your stomach has about a 1 to 2 pH value. Okay, so when we talk about, all right, acids and bases, we're going to discuss these three things. Bicarbonate ions. If you remember, we were talking about in the circulatory chapter, we talked about what bicarbonate is. It's a buffer, right? And buffers help to keep the pH value, all right, of your blood in that 3.5 to 7, excuse me, that's 3.5. 7.35 to 7.45 range. That's what the buffers do. So if you start to become too acidic, it helps to decrease that, or actually increase the pH value. Okay? Hydrogen ions, when you see hydrogen ions, that's this, H+. And you should think acidic. The more hydrogen ions, the higher concentration of hydrogen ions, the more acidic something is. So there's an inverse relationship. If you increase hydrogen ion concentration, that's what these brackets mean, concentration. You increase the hydrogen ion concentration in a solution, all right, you will decrease the pH value, which means you'll make something more acidic. Okay? All right. So what we're going to see here, all right, we're going to see these substances, bicarbonate ions, hydrogen ions, are going to move freely right across our membrane there, our filter. Okay? Important concept. We do not resorb bicarb. That's what this is. That's bicarbonate. HCO3. That is called bicarbonate. You'll hear me refer to it as bicarb. All right? We do not resorb that. It gets filtered out. It doesn't come back in. all right? We reclaim it through a process that I'm going to go over with you here, all right, in which we will replace the filtered bicarbonate with a brand new molecule of bicarbonate. Okay, so you really have to understand, all right, that this fact here, that it is the filtered bicarbonate is not resorbed. We do not resorb that. All right, we're going to make it. Okay, so I'm going to go over that with you. Okay, so really where we're going to see, all right, the end product here in the urine, what really is going to affect the pH of the urine here is what is going to happen here in the collecting tubules. Okay, so determining whether or not the urine is going to be acidic or basic, all right, and the blood's going to be acidic or basic really comes down to what happens here in the collecting tubules. All right, so here's what's going to happen. If your blood is too acidic, right, what did I say? We need to buffer it, okay? So if something is too acidic, that means we have way too many of these things floating around. We got too many hydrogen ions floating around. So we need something that's going to go in there and it's going to suck all those up like a sponge, okay? So what are we going to do? We're going to make more bicarbonate here, all right? So we're going to make more bicarbonate, we're going to synthesize it, and then when we make that new bicarbonate, it gets resorbed into the blood, all right, and then it's going to hook up with the hydrogen ions, and when it hooks up with the hydrogen ions, it's going to it's going to decrease the hydrogen ion concentration, but at the same time, it's going to increase, all right, the pH of the blood. All right, I'll go through all this here with you in a second. If the blood is too basic or alkaline, all right, then what we're going to do is we're going to secrete bicarb. We're going to put it into the urine, get rid of it, all right, and we're going to resorb the hydrogen ions. Because remember, hydrogen ions will increase the pH. All right, so if we secrete the hydrogen the bicarbonate in the urine, all right, and we resorb hydrogen ions into the blood, all right, we are going to lower the blood pH because it's becoming more acidic, and we're going to increase the urine pH because we're putting more bicarbonate into the urine, and that's going to make it more basic. I'll show you how all this works, all right? But you do need to know the cell types. So acidic blood, easy to remember. We're going to deal with type A intercalated cells, all right? A acidic, type A, all right? And then if our blood is too basic or alkaline, I just remember basic, B, all right? We're going to deal with type B intercalated cells, okay? All right. So here when we're talking about the movement of bicarbonate, hydrogen ions, all right, we're going to see, all right, a lot of that bicarbonate is going to be, look at the term, reclaimed in the proximal convoluted tubule. It won't be resorbed, all right? Again, some down here in the nephron loop, but a lot when we're dealing with, all right, the acidity of the blood and the acidity of the urine is going to be regulated here in the distal convoluted tubule, the collecting tubule, and the collecting duct here. All right, so let me go through and tell you the story here as I take a sip of water. All right, let's start at the beginning. In the proximal convoluted tubule, in the area in which we're going to reclaim, all right, our bicarbonate. Okay, so this is what is going to happen. And you've seen some of these equations before, so this is not going to be too new to you. Whoops. Okay, so here we'll make that a little bit smaller. Okay, so here's our tubular fluid, okay? in our tubular fluid, we've just pushed out of our blood bicarbonate and hydrogen ions. Okay? So what will happen is, for a brief moment, the bicarbonate and the hydrogen ion will combine, all right, to form carbonic acid. It's brief because carbonic acid is not a very stable molecule, which is okay. That's fine. All right? We don't want it to be too stable because stable molecules are harder to break apart. So it's relatively unstable. So to briefly combine into carbonic acid. And then that carbonic acid will break down to or convert into carbon dioxide and water. I'm cool with that. Remember we saw that in the red blood cell when carbon dioxide wanted to enter into the red blood cell, the chlorine shift. You guys remember that? Okay, when we're talking about the buffering ability of the blood. Well, this is what we're going to see here. We're going to need that carbonic anhydrase, which is that enzyme that helps to facilitate this process. So here's what happens, right? We now have carbon dioxide and water. Well, guess what? Carbon dioxide is a respiratory gas. It can diffuse directly across, all right, a plasma membrane. It doesn't need any help from any type of channels. So it goes into the cell. All right, boom. Water, it stays outside. Okay, it's going to stay in our tubular fluid. It's not going to follow carbon dioxide. There's not going to be an osmotic component for that. So carbon dioxide enters into the cell, all right, which it will recombine with water. It'll give us carbonic acid briefly, and then carbonic acid, all right, will break down into the bicarbonate and that hydrogen ion. Okay, the bicarbonate then moves across, all right, the basolateral membrane back into the blood. Okay, so it's a whole new, this molecule is different from this molecule. You see what I'm saying? So we don't resorb it. We broke it down, did some stuff to the molecules, and then we made a brand new molecule. So we can't technically say that we resorbed it. All right, when glucose gets filtered out of the blood, we don't break the glucose molecule down and rebuild it back into the cell. That same glucose molecule goes from the blood into the tubular filtrate, all right, and then back into the blood through the cells, but it never gets changed in any way, okay? This molecule does. The bicarbonate gets converted into the carbonic acid, then it gets converted into carbon dioxide and water, and then it just change. Then it'll recombine and make a whole brand new molecule. That's why we say, all right, that it's not resorbed, okay? So it's replaced essentially. We replace this bicarbonate molecule with this bicarbonate molecule and dump it back into the blood. Meanwhile, all right, when carbonic acid, all right, is broken down into bicarbonate, that hydrogen ion is pumped out of the cell and back into the tubular fluid. So guess what we're doing now? We're starting to increase the concentration levels of the hydrogen ions in our tubular fluid. So what does that mean? The tubular fluid is becoming more acidic. All right, that means the pH value is decreasing. Okay?
What's decreasing?
The pH value, okay, in the tubular fluid. So that means just becoming more acidic there. All right? So what we're seeing here is this is what is always happening. So our net effect, all right, as we quote unquote reclaim the hydrogen, the bicarbonate, all right? Again, it's a different molecule. This is different than this, all right? But we're replacing one bicarbonate molecule for another bicarbonate molecule. That's all we're doing. But at the same time, we're going to lose, all right, a hydrogen ion into the tubular fluid, all right, which essentially will eventually become urine, okay? So that's where, all right, the lowering of the pH of the urine occurs, making the urine more acidic, okay? Or at the same time, as we put bicarbonate back into the blood, all right, if there's any hydrogen ions floating around in the blood, all right, then the bicarbonate is going to do this, what we just saw here. This same process will occur in the blood. Yeah, it'll reattach itself to a free-floating hydrogen ion here in the blood, but thereby, if we will thereby reduce hydrogen ion concentration in the blood, And remember our equation, right? If you reduce hydrogen ion concentration, you're going to increase the pH value. Well, don't think it's not. We're not trying to balance the urine with the blood pH, but what we're going to see is it's just going to think of it as a sponge. This is a sponge that's going to go around looking for hydrogen ions, and it's going. And when it does, when it finds it, all right, it's going to undergo this chemical equation here, this process here. And what's our end result? Well, look at our products: carbon dioxide and water. Carbon dioxide, those are two inert, I shouldn't say inert, but they're two harmless substances in the body. All right, questions about that? All right, so now let's talk about what happens, right, when the blood is too acidic. When we're down here, all right, in the collecting duct, the collecting tubule, and the distal convoluted tubules. Now we're going to be dealing with the intercalated cells here. All right, so all right, so now we're going to see here, all right, in the intercalated cells, depending on whatever is going on in the blood, all right, what if the blood is too acidic in this situation, all right, that means our blood has tons, all right, of hydrogen ions floating in it, all right. So if the blood is too acidic, we have to buffer it. How do we buffer it? We need to make this. Okay, so how do we do that? We just saw it on the previous slide. All right, our cells, these type A intercalated, are going to just absorb and take in as much carbon dioxide that they can from the tubular fluid. Okay, it'll enter into the cell. That carbon dioxide will combine with the water and give us the carbonic acid. Carbonic acid will then break down into bicarbonate and then the hydrogen ion. So the hydrogen ion gets secreted, all right, into the urine, decreasing the pH. Now the urine is more acidic. And this bicarbonate moves into the blood and it looks for these guys. And when it looks for these guys, it will combine with them and eventually give us carbon dioxide and water, thereby increasing the pH of the blood. That's what we want. Okay. Done with resorption. Whew, that was just resorption. I know. I mean, after this chapter, you can appreciate your kidneys a little bit more. All right, yeah, we know they're filtering, but look at all the stuff that they do and how much they keep everything in balance. It's a lot. Okay, but speaking of a lot, what happens if you have a lot of protein? You're a person that likes to consume twice their body weight in grams of protein. That's a lot. All right, we break proteins down into amino acids, but part of that process is going to give us, all right, what we call nitrogenous wastes. All right, and nitrogenous wastes are going to yield us this molecule here, urea. So as we break down proteins, we're going to increase the amount of urea that we produce. Okay, and urea, all right, contains nitrogen and some hydrogen atoms into it. Okay, so with urea, all right, what we're going to see is we're going to see both secretion and resorption, all right, but about 50% of it we're going to get rid of, all right, in our urine, which is good. That's a very good percentage here, all right. But part of this, all right, ability for us to get rid of urea is that we have to establish a concentration gradient, all right, because then basically think of it like this. If you don't have a concentration gradient, okay, you're not going to see certain processes, all right, passive processes work because you need a concentration gradient, all right, because you need something to move down its concentration gradient in a passive process. Active processes is a different story. When there's a concentration gradient, we're going to usually be moving things against the concentration gradient or up, okay? But if we don't have a concentration gradient, our passive processes, diffusion, facilitated diffusion, and osmosis, we're going to have issues there, okay? So we're going to excrete our urea, all right, in our urine, all right. About 50% of the body's amount of urea, all right, will be gotten rid of in the urine. All right, you have a very small amount that actually is gotten rid of through sweating. Okay, uric acid. All right, this is what happens when we break down our nucleic acids. All right, nucleic acids are found in our RNA and DNA, for example. Okay. So we'll also see resorption and secretion. That, remember this guy, creatinine? All right, for muscle metabolism, okay, we had the phosphate system in which we basically, when we're dealing with creatinine, we're going to just transfer our phosphate from one molecule to another molecule. And so that system here helps, all right, when we're dealing with skeletal muscle metabolism, not just skeletal muscle, all right? But this is only secreted. So creatinine is actually a good substance to use to monitor, all right, the actual how kidneys are functioning. Okay, if we have time, we'll talk about that. All right, what else do we get rid of with the kidneys? All right, drugs. All right, tons of drugs. All right, anything that you're going to be taking into your body that you have to activate, you'll get, especially the drugs. All right, depends on first pass, second pass. All right, we'll get rid of drugs in the kidneys. Remember when we talked about red blood cells and we broke down red blood cells and we recycle a lot of the material? All right, well, hemoglobin during that process of getting broken down, all right, gets converted into urobilin. All right, well, there's urobilirubin. All right, this is one of the intermediates. All right, but it's this urobilin that gives your urine that yellow coloration there. All right, and then of course some of the hormones will be eliminated through the kidney. Okay, this one should look familiar. HCG. That's the hormone, all right, that a pregnant female produces, and that's the hormone that is what a pregnancy test detects. All right, and then if you don't have enough adrenaline, no problem, you're peeing out some of it anyways. Okay, so we've talked about concentration gradients up to this point, so I want to talk a little bit about how we create a concentration gradient, and this is kind of where we're going to be talking about the nephron. All right, and when I said we've been talking a lot about the nephron, but we're really going to get into the nephron loop. That one structure, you have the proximal convoluted tubule, and then it takes this big dip down and it comes back up. We're really going to be talking about the nephron loop here. Okay, so when we're talking about our concentration gradient, we're going to be discussing, all right, the interstitial fluid. That's the fluid that's going to be surrounding the nephron. That's going to be our two reference points. Basically, everything that's in the tube and then what's outside the tube. Okay? So this concentration gradient, all right, relies on the presence of certain solutes. All right, big time with sodium, all right, potassium, depending on what we're talking about. Well, sodium and chloride together form salt. That's going to play a big role, especially when we talk about the movement of water, all right, with osmosis. So a big point that you want to understand when we talk about the concentration gradient, right, this is an important fact. It will progressively increase when we go from the cortex into the medulla. Now, what the heck does that mean? Well, you remember the cortex of the kidney and the medulla of the kidney. The cortex is the outer portion, the kidney is the inner portion. Well, parts of these nephrons, all right, those nephron loops, all right, the majority of them will be in the cortex, but part of them will dip down into the medulla, all right? So certain parts of that nephron loop will be found here in the medulla, and that medulla is going to have an increase in the concentration gradient of whatever we're going to be talking about here shortly compared to parts of the cortex there, all right? So when we're dealing with water, osmotic pull plays a huge role in that. All right, so that's what we're going to be talking about here. All right, we're going to talk about how this concentration gradient is going to affect the movement of water either into the interstitial fluid or out of the interstitial fluid. And of course, all right, when antidiuretic hormone is present, its effects on that. Okay? All right. So now we're going to deal with, we've talked about the proximal convoluted tubule, the distal convoluted tubule. We've talked about the collecting tubules, the collecting duct. We really haven't talked too much about the nephron loop. All right. Again, that nephron loop is that big loop that just drops down from the nephron here. Okay. So we're going to talk about two concepts here when we're dealing with the nephron loop. The first one is a positive feedback mechanism. All right. You remember positive and negative feedback. All right. Negative feedback is that the outcome of whatever it is that we're trying to affect will always be in the opposite direction of the stimulus. Perfect example, body temperature. If you get too hot, what does your body want to do? Cool you down, turns your body temperature down. All right. In this situation, we're going to deal with a positive feedback. All right. Positive feedback is going to further promote whatever that stimulus is. All right. So for example, if you have a lot of calcium in your blood, now we're going to put even more calcium in your blood. I'm not using that for this example here, but I'm just kind of giving you, all right, breastfeeding is another example, all right? Milk production, all right, baby is suckling, more milk is produced, all right? Well, this is what we're going to see here with what we call the countercurrent multiplier. And this is going to help us get our concentration gradient in effect, okay? So remember these types of nephrons, the juxtamedullary nephron? Juxtamedullary nephron was that nephron that was right there at the border of the cortex and the medulla of the kidney. All right, so again, the majority of the nephron was in the cortex, but that nephron loop dipped down deep into the medulla area. All right, so couple things to review, all right, from lab. In our loop, you had the descending limb. That's this part that goes down. That's the descending limb right here. Okay? Put this in your memory banks. Your descending limb is permeable to water, but impermeable to salts. Okay? So water can move in and out of that, but the salts cannot. What's the salt? Sodium chloride. Okay? So that means that water can go from inside the tube, the tubular fluid, and it can leave the tube. So it's going to exit out. So here's our loop. All right, water comes out of it. Okay? Meanwhile, salts stay inside. So if water leaves the tubular fluid, all right, and salts are left inside, does that tubular fluid become more concentrated with salt or more dilute? What would you say? Water leaves, salts are left behind. Is it more concentrated or more diluted? More concentrated. Right, right. So we're going to increase, all right, that salt concentration in the tubular fluid. Okay, so we're more concentrated here. Now, the ascending limb, that's the part of the loop that goes upwards. Okay? Again, put this into your memory. All right, we're just going to flip it. That part becomes impermeable to water. Salts get pumped out. They leave. Okay? So the salts leave, water stays. So what does that mean to the tubular fluid there? Does that become more concentrated or less concentrated? That's right, less. Okay, so it's getting more diluted because water, all right, stays in. It doesn't go anywhere. Salt leaves, so more water is left behind, so it becomes more diluted. Okay? So I got a good picture here that I'll show you. Okay? So the more concentrated the salts are, the more gets pumped out. All right? Obviously, if it's less concentrated, less gets pumped out. So here we'll see our salt concentration gradient. All right, it's all about creating these concentration gradients here. I'm going to come right back to the slide here. So don't pay, well, I'll just zoom in so you can pay attention. Okay, so here's the counter current multiplier. Here's our tubular fluid here. Right? Here is the blood vessel, the vasa recti. Okay, so you can see when we're talking about milliosmolarity or osmolarity, okay, what that means is how concentrated, all right, the fluid is. So I don't expect you to know these values, but I expect you to know this. Is this value smaller than this value? Okay, the larger the value, the more concentrated something is. Okay, that's what you need to know for that. We're talking about osmolarity. So this is pretty concentrated. This, not so much. Okay, because water's in there to dilute it. Okay, so when we come down, this is the descending, all right, limb of the nephron loop. Okay, it is impermeable to salts, but permeable to water. So water leaves. And so as the fluid moves through this, water leaves. It goes into, this is the interstitial fluid, right? This blue stuff. Right? It leaves and it goes into the interstitial fluid. As we go further, is it becoming more concentrated the more we go down, would you say? Yeah. Okay. So as we get down here, all right, that tubular fluid is really, really concentrated. All right, a lot of the water left. Okay. Salt couldn't leave. Okay, that's okay. That's okay. So now we get here now into the nephron loop and the ascending limb. As we start to ascend on this portion, okay, the ascending loop is impermeable to water. So water can't get in, all right, but salt can leave. So what happens? Salt leaves because, all right, it's going to go out, it's going to go down its concentration gradient. It's going to go from an area of high salt concentration to low salt concentration. So it leaves. And then the only thing that's left behind is a little bit of salt and the water that stayed in there. All right. And now as we go from here up to here, you can see we become more dilute. All right. So again, all this is I'm trying to show you is that we're creating a concentration gradient, which means if we have a concentration gradient, we can have things move. Okay. And we can tell which stuff can't move because if it's impermeable, it's not leaving. Okay? The only thing that can leave is whatever is going to be permeable to that area there. All right? So that's the counter current multiplier there. That's all I'm just trying to show you. All right? What we can see leaving. Because now we've got another structure or another concept that I want to talk about, which is called the counter current exchange system. This is different. All right. So staying on our subject of concentration gradients, all right, the vasa recta, again, is going to be that capillary system that is going to surround the nephron loop. But in this case, all right, the blood flow is going in the opposite direction of the tubular fluid flow. All right, so what does that mean? You can see here. All right, the tubular fluid is going from here to over here. All right, the blood flow, though, is going in the opposite direction. Okay, so tubular fluid movement is going from left to right, like reading a book. Blood flow is going from right to left. All right, so that's what we call it, counter current exchange there. So what's going on here? Okay, again, this is all about concentration gradients, folks. All right, but since we're dealing with just water, we're now going to see, all right, what's going to happen with water in the blood vessels. All right, our previous concept we were talking about what's going on with the tubular fluid. Now we're going to talk about what's going on with the blood. Now we're going to see what the water is going to do, all right, in regards to where it is in that capillary bed. And that's all operated by osmosis. Okay? Salt, all right, or our solutes, all right, that is going to move, all right, or be affected by diffusion. Okay, remember, if it's osmosis, the only thing we're talking about moving is water, nothing else. Okay, water is the only thing that is going to be moving with osmosis. Okay, diffusion is when we get the movement of solutes. All right, and our solute here is going to be salt. Okay, so in this situation, when we're talking about the countercurrent exchange system, we're going to see how water is going to move out of the capillaries and how salt is going to enter into the capillaries. Okay? So as it enters into the capillaries, doesn't it make sense that it's going to increase the concentration inside the capillaries? All right? So we're going to see here, all right, I'll show you on the slide here in a moment, all right, what's going to happen with our concentration gradients when we go from the ascending limb to the descending limb or vice versa. All right, let's go back to our picture. Pictures are better. Okay, so again, if you look here, now we're dealing with the blood system, okay, the vasa recta. Here again is the osmolarity, how concentrated it is, and you can see, all right, up here, not terribly concentrated. Down here, we're seeing a lot more concentration. Now we'll move in the direction of flow. So over here on the right side, all right, so as our blood is moving through, again, in the opposite direction of the filtrate flow, we're going to see, we're moving down, or we're going, as we move down the vasa recta here, we're increasing our concentration of salts, all right? So what will happen is water will exit, because remember, it wants to go to where there's a higher salt concentration. And as that's occurring, all right, our salts will enter in. So water exits, salt enters in, okay? And then vice versa, as we go through the descending limb region here, okay, as we're going from a higher concentration to a lower concentration, all right, water will enter back in, okay, because of the concentration is so high for the solutes, and water always wants to follow where there's more solutes, all right? So as it follows where there's more solutes, it's going to be where there's less concentration of water. So water is going to flow down its concentration gradient, and then the salts will exit. It can be somewhat complicated, I get that. And most times it takes a little bit of while to understand that, but just trust me, you know, just trust me. It's tough to throw it out there, yeah. Okay, let's take a break. You folks have been awesome and listening to me ramble on for the past hour and 25 minutes. Let's break here for a moment.